Hi everyone, welcome to the 7th RI session. So my name is Ina Sophia and I'll be your host for today. So as everyone might be aware, this session is broadcasted across MBIO's Facebook page and also on this Zoom session. So I really appreciate it if everyone could turn off your microphone to avoid any interruption during the session. And also feel free to share the link to your friends or your family. And yeah, if you want to share this session with others. Apologies if there's like background sound, so sorry about that. This RI session was not made possible without the generous sponsorship from our gold tier sponsor, UK Education Centre, UKEC branch. UKEC is the leading education agency in the UK that provides free support and services for students who are planning to pursue their higher education in the UK. Some of these services include university application and visa application. So if you have any of your friends or family members who are interested in applying for UK university, I would definitely recommend for you to apply through UKEC because they will help you tremendously with the application process. So I would also like to take this opportunity to introduce to you the MBIO certificate program. It's on the slide here. So this scheme allows MBIOS members to get certificates upon their participation in any of the MBIOS events. And as you can see there are three kinds of certificates we offer, which includes like gold, bronze, and silver. So it would depend on the number of events that you join. You will get different kind of certificate. And this certificate will be particularly helpful if you put on your CV or you post on your LinkedIn or something. So yeah, I would definitely suggest for you to join more MBIOS event and screenshot as an evidence. And then you can send the evidence to us and claim the certificate. So so now I would like to introduce our speaker for today, Professor Christina Chai. So Prof. Christina completed her bachelor's degree in chemistry at the University of Canterbury, New Zealand. Then she completed her PhD in chemistry at Australian National University, Australia. And Prof. Christina is a very reputable lecturer with more than 20 years of experience. And currently she is a professor and the head of the Department of Pharmacy at the National University of Singapore. And today, Professor Christina will be sharing about the trials and tribulations of natural products, drug discovery and development. She will also share more about the research that she's actively involved in related to treating various diseases with natural products. So without further ado, everyone, please join me in welcoming our wonderful speaker, Professor Christina Chai.
So thank you everybody for actually turning up, okay? And I know you're all in some weird time zone and it is a Saturday. So when I was younger, Saturdays was really not listening to seminars. So I really would like to say that I am incredibly impressed that you even turn up. So also, I would like to thank MBIOS for this opportunity to give a talk today. It won't be very long. It'll just to give you a kind of idea of the sort of research that I am doing. I started with this title, which is Natural Products in Drug Discovery and Development. But it could alternatively, my other choice was to give a general talk, which I would have given a talk something very similar to this title, which is There and Back Again, A Scientist's Tale, which is by me, okay? And that's really because of the fact that, like a lot of you guys, you know, we have traveled around the world. So I started also as a Malaysian who did a year high school in Wellington, New Zealand. And after that, I went to do my undergraduate studies at University of Canterbury in Christchurch. That's pretty cool, okay. And from there, I got a scholarship to do a PhD at the Australian National University. And then I did a year postdoc at Oxford University in the UK. And for those who are in the UK, you will know that it's really quite expensive. And as a postdoc, I was getting paid peanuts. And I was like thinking, I don't know how long I would be able to tahan this hardship, right? So in the end, I applied for a position, which was my first academic position, and that was back in New Zealand, and that was to Victoria University of Wellington. Now, I must say that I never really considered whether I wanted to be an academic, but out of sheer desperation of trying to get out of UK, okay, to somewhere a little bit more reasonable at a time, in my view. So that's why I took up my first academic position. My second academic position was back in my alma mater, which was at ANU in Canberra. And for a lot of reasons, I gave up my tenured position over there and I moved to Singapore. I didn't join NUS to begin with. I joined a research institute because at a time when they recruited me, they told me, you can establish a research program and you can have as much money and you can have as much resources as you needed. So that was something that was really tempting for me. And that's how I landed in Singapore. But then I realized that academia is really more suited for me than any other position, which in that position, there was a lot more administration, relating to setting up research and a research program. I'm happy to share a little bit more if there are questions at the end. So your questions need not be limited to that related to science. So I'm going to start talking about the area of my research. But firstly, I'd like you to think about what your parents actually tell you to take, okay? For example, when you have a sore throat. And more likely than not, right? They will tell you, okay, drink honey, okay, with lemon, okay? And what would they tell you when you say you are feeling tired? They will say, why don't you take some ginseng, okay? Or what happens when you say your eye is feeling very tired, it's a little bit sore and blurry, they will say, okay, probably you need to take some goji berry, put it in some hot water and drink the water, okay? And in the time of COVID, what did they tell you to prevent getting COVID? And those in the UK, that there was very little control. So maybe you should have listened to your parents. They would probably have said, you should basically eat a lot of turmeric, okay? And that will prevent you from getting COVID. All these examples that I show you really are related to products from natural sources or natural products. But the question is really, what our parents tell us, is that real or is that like really quack? Okay, because especially mothers, they are the worst quack doctors ever, right? They're always telling us, based on zero scientific knowledge. But there must be some evidence of truth that they would have read somewhere. But to uncover the real truth, you and I must actually understand and evaluate the scientific evidence. The problem is that scientific evidence tend to kind of contradict themselves, right? So sometimes literature is not experimentally convincing, or it's flawed in design, or they are not reproducible. And this is basically why, in my view, public doesn't really trust scientists very much. So let me just go back to this definition of natural products. It's really more than the things we ingest, okay? They come from a wide variety of sources: plants, microorganisms, marine organisms, and actually they are secondary metabolites. Primary metabolites are those that is needed for the functioning of the organisms, but usually the secondary metabolites, the sheer fact that they are produced, people don't really know what they are really used for that organism. But from a medicinal chemistry point of view, right, they are a rich source of potential drugs in drug discovery and development. I'm going to show you here the two chemical structures, if you remember your chemistry from your way past, okay? But if you look at the structure of Taxol and the structure of artemisinin, they are two very, very good examples of natural products which had actually been used as drugs. So Taxol is a cancer chemotherapeutic. And if you look at the structure of Taxol, if you were a synthetic organic chemist, you would never in a million years try to make it. It's so complex. There's so many chiral centers, but yet it is produced by a plant, and this is isolated from the bark of a Pacific yew tree. So you can see the bark over there. But you know what the problem is? In order for you to treat one patient, okay, one treatment, one patient, you need to harvest 6 of these trees, but they take 100 years to grow. So although the anti-cancer activity of Taxol was known for a very long time, it never really made to go very far because there was limited in resource. Now, okay, Taxol is not isolated from the bark of the Pacific yew tree. So it's actually, they've developed organisms which would be able to produce advanced intermediates of Taxol, and then you can do a chemical modification. And they also found that a very common European yew, from the needles, the leaves, you can actually isolate the right-hand portion of the molecule, and then you just need to do a very simple derivatization to be able to get Taxol. The other example is this compound known as artemisinin. The lady who actually discovered it as an anti-malarial won the Nobel Prize in 2015, if I'm not mistaken. Artemisinin is isolated from this Artemisinin annua, okay? And actually, this has been known for a very long time. But as a synthetic organic chemist, you will, again, never think in a million years you will make this sort of compound. Because if you don't know, I'm telling you now, those sort of bridge where the 2 oxygens are present, that's an endoperoxide. And actually, peroxides, I will never let my students work with peroxide because they are potentially explosive. So this is kind of really interesting, right? How natural products tell us that these compounds, you can make it, it's stable. And actually, it is, and it has potential therapeutic applications. In actual fact, this is a question of debate for a very long time. In the very early days, when science wasn't very advanced, there was no real equipment, and you can only guess what is present in the natural product, okay? People moved away from natural products to look at synthetic compounds. In other words, you make compounds and then you test them. If it works, it works. If it doesn't work, it doesn't work. So then there was a move away from people looking at natural products, okay? So there's this debate of whether drug discovery and natural products is really the end of an era. That means nobody should really worry about looking at it, or whether it should be an endless frontier for which you should be able to get new drugs for diseases. So if you look at the paper shown at the bottom over here, this is data that was taken from these 2 people who actually really try and find out, looking at literature, the small molecule approved drugs from 1981 to 2019. And all you need to do is actually look at the top half. N is natural product or natural product derived. S star just means that the pharmacophore, which is the portion that actually is present in the natural product, okay, is basically present in all of these FDA small molecule approved drugs. So when you kind of look at this, right, you know that really a little bit less than 50%, but pretty close to it, actually are natural products or natural product derived or natural product mimics. So this tells us that you shouldn't basically be giving up on natural products, because if you look at history, it is the source of quite a number of good drugs. And when you think about this, right, it kind of really makes sense. So on the left, if you look at the synthetic compounds, let's say you say, okay, let's collect all the synthetic compounds you can ever make. That could be 22 million. But if you are trying to look for something which is biologically relevant, then the reality is that you're almost looking for a needle in a haystack, right? You're going to start from a massive library and you're going to find something which is going to be biologically relevant. But on the right-hand side, if you look at starting with natural compounds, you know they're going to start by being biologically relevant. And And from there, if you start looking at this very so-called relatively small library, the chances of you being able to find something is in fact going to be quite high. So now we come back to this issue associated with natural products and therapeutics. And of course, we are all very familiar with traditional medicines. Okay, if you think about the traditional Chinese medicine, the traditional Malay medicine, Sabah, Sarawakian, the indigenous, the traditional Ayurvedic medicines, all of that is really knowledge that is passed on, okay, and it's part of history and culture. But the problem is that, do we really know if it works, okay? It's not really usually just one compound, but there are usually multiple components that are going to be present. So let's give some examples over here. So the bottom over here, you can see there are a number of Chinese herbs, okay? And the one at the top with turmeric, okay, that's really quite common these days. And if you look at that, you can see that on the chemical structures that are shown are the main chemical compounds that are present in those herbs that are thought to be responsible for the biological activity. And if you think about this and you look at some of these, and I've just isolated just 3 compounds over here, you can see that it's like a magic thing if you truly believe it, right? It's like, oh, if you take resveratrol, which for example is present in red wine, okay, it's supposed to be anti-cancer, neuroprotective, cardioprotective, blah, blah, blah. Okay, so when something is good for everything, is it really true? Is it fake news, or is it truly good for nothing? So I think this is a very interesting area of research. Although I have looked at studied natural products for a very long time, I kind of feel a little bit uncomfortable thinking that you've got a compound that is potentially going to be able to cure you for every conceivable disease that you would get. And this is basically the basis of my interest in natural products. In actual fact, there is also the other problem. And this is a problem about oral bioavailability. So if you are a pharmacy student, you will know what I'm talking about. It's really about when you ingest something, how much of it really gets into the organs or into the organs in your body that is required for the biological activity. And the reality is that if you hear people tell you, okay, you should drink green tea is fantastic for you, it's an antioxidant, all this other stuff. The reality is you gotta drink a heck of a lot of green tea, right? Because the oral bioavailability of the major component in there, which is EGCG, is 0.1 to 0.3%. And in the case of curcumin, you know if you have started taking it, it's not soluble. It's a yellow suspension, because I see my cousins drinking it and I'm like, oh my God, that cannot be nice, right? And the bioavailability is only 1%. So again, you will need to consume quite a lot of it to be able to get enough to be able to give you the biological activity that has actually been reported. So what is the truth? I really don't know, but the truth is actually out there. And I feel that in view of the huge potential of natural products in drug discovery and development, that this is something that one would need to actually study. So let's go back and ask this very scientific question, right? If you look at a natural product and they have a lot of different sort of biological activity, is it because they are promiscuous? So let's talk about promiscuity. Promiscuity, if you look at the definition, it actually implies that it's not selective, it's indiscriminate, okay? And in other words, what we're trying to say here, right, is natural products just going to react with a whole bunch of targets because they are promiscuous? And does this actually make sense? So let's think about this, right? Natural products are biosynthesized, okay? When they are biosynthesized, it goes through a whole bunch of enzymatic processes to give you the product at the end of the day, which means that it is not surprising, okay, that they would and they could actually have multiple targets. This question about multiple targets actually is it's a bit anti what we have been taught in terms of drug development. Because if you look at pharmaceutical companies, the original paradigm has always been that you create a drug that is selective. Okay, so this is based on Paul Ehrlich's magic bullet. So you got a single target paradigm. So if you're going to be able to target that one target, and you will be cured of a disease. The problem with this is that now people know that actually there's a lot of diseases that need complex cures. So actually you need a drug that will target multiple targets, which additively will give you a therapeutic effect, okay? Not just have one target, but which, for example, is to cure Parkinson's disease, etc., etc. In other words, you are looking for drugs which have polypharmacology. So in essence, the reality is the paradigm shift from a single target to a multi-target paradigm. So it's like shooting in all directions, right? The only problem with this is that as a pharmaceutical company, and even in academia, this is really quite hard to be able to study, because you're looking at which of the targets are going to be relevant to the particular disease area. So let's go back and talk about promiscuity in natural products. Actually, there's lots and lots of examples. So I've just given 3 examples over here. Aspirin, which is a derivative of salicylic acid, it's a prodrug of salicylic acid, okay? And actually, it's got multiple targets and multiple modes of action. Artemisinin, which I mentioned, has been shown to have a lot of protein targets, 124. And the one that's shown, flavone, apigenin, okay, is also known to have a lot of targets that are possible. When you look at these structures over here, you would see that there are, in fact, some sort of common sort of features. So they could have promiscuity. In other words, they could be multi-targeting because of the pharmacophore, the pharmacophore being the unit that is responsible for biological activity. So if you have a common pharmacophore, like the one in aspirin, right, you are not surprised that that pharmacophore will interact with a number of targets. Or you could have reactive functional groups like artemisinin, because I said it's an endoperoxide bridge. Okay, so it's going to be very reactive. Okay, so when it's very reactive, you can imagine that it will be able to interact with sustained functionalities in multiple sort of protein targets, for example. And then you've got functional groups with multiple reactivities. For example, apigenin. You've got hydroxy groups which are capable of hydrogen bonding. It would be capable of redox cycling. It would be capable of metal coordination. And maybe that accounts for its promiscuity. The reality is that this is something that I feel that is very interesting to study. I'm a medicinal chemist. Actually, I'm a synthetic organic chemist that when I moved to NUS Pharmacy, I moved my research directions to that of medicinal chemistry, and I find it very interesting. I have, thinking from a chemist's point of view, I challenge a lot of things that have actually been accepted by the pure medicinal chemists, okay? So here are the possible things that, that could result from polypharmacology, and there's a compound that I've shown on the right-hand side, and I'm going to talk about this. And this compound is known as andrographolite. So this one comes back to this herb. And I gave this talk in Nice last year, and quite a number of people basically know this herb very, very well. Actually, this herb is from a plant which is known as Andrographolis paniculata. But there are different names for this depending on where you are. It's actually one of the World Health Organization recognized herbs, okay? It's present in traditional Chinese medicine, Ayurvedic medicine, and some Western medicines as well. And this is the active component in there. And you know, when the time in 2020, when we're all so desperate for a cure for COVID, and I know, for example, my relatives in Malaysia, they were telling me, oh, you know, the person who sold the vegetable at this corner of the shop, he died of COVID. The other one who did the char kway teow also died of COVID. So there was a lot of fear, right? So this was what was published in the Straits Times in December 2020. In Thailand, they cleared the use of this herbal medicine, which they call, which is the same, Androgynophalis paniculata, or called green chirita, okay, as an alternative medicine for the treatment of COVID-19 antiviral. But it's especially before COVID, it was especially known for its anti-inflammatory properties. So it's for the treatment of like respiratory problem, okay? And when you talk about this, remember, I'm talking about multi-targeting and promiscuity. Certainly, andrographolide is very promiscuous. And, you know, they thought that the reality is that it is going to bind to this transcription factor, which is known as NF-kappa B, and the biologists would know this very well, which is a master regulator. And then that's why it has all these wonderful effects, because that has downstream effects which will affect neurodegenerative disease, antiviral, etc., etc. And there is, in fact, some evidence that shows that it actually covalently binds to the NF-kappa B. Then it has also been shown that it also binds to another protein which is involved in upregulation of antioxidant enzyme. And in the context of antivirals, that is thought to be binding to a protease, okay, which is then going to affect the survival of the actual virus itself. If you read the literature and if you 100% believe the literature, you will not do any more research, okay? So I tell my PhD students, you have to be critical, okay? Because, you know, you almost cannot believe, you gotta look at the data. So actually we looked at the data and we started out by asking this question about trying to understand which is the part of the molecule that is really required for the biological activity. And we did some really interesting stuff. And in view that this is a general audience, I'm not going to belabor the structures, but I'm going to say that we varied the bits that circle, the bits that's basically rectangular. We tested the compounds and we found some very interesting things. One of the key things that we found was that NF-kappa B is not really the mechanism for its anti-inflammation action. You see the studies that I shown before, where they have actually an adduct, is because they just basically took andrographolide and they incubated it with NF-kappa B. Okay, but that doesn't mean that in the body, that's basically the target. So what we had done was to look at andrographolide along with another natural products, made a whole bunch of synthetic compound libraries, and then we actually discovered and patented quite a number of the compounds associated with this. So the top one had a different mechanism. It's got anti-inflammatory activities as well. The bottom one was not really very as active as andrographolide, but it's really going to be less toxic as well. So now let's talk about the antiviral activities, because I'm sure that you want to know whether you should go and buy andrographolide pills, right? Okay. The reality is that the antiviral activity has been reported for a very long time, okay, against a whole bunch of viruses, which is kind of also a little bit weird. And before COVID-19, right, we were working on chikungunya virus. It's I'm not sure whether you know chikungunya. Anyway, it's not a very severe disease in terms of it's not at the pandemic stage, but my colleague in microbiology reported that andrographolide was effective against chikungunya virus at 77 micromolar, which is really not very active. And of course, this is the paper that was about Androgynophalis paniculata extract and anti-SARS-CoV-2 activity. So we started the work on chikungunya viral infection because remember, we have done a lot of work on anti-inflammatory compounds and we now have a library of compounds. And what we discovered was that we can actually make compounds which look like andrographolide, the natural product, but not quite, okay, which are going to be much more potent than the natural product itself. Then you could ask, like I asked, hey, are you sure this is not really related to the anti-inflammatory properties? And indeed, you know, the compounds that were active very active against viral, in this case, chikungunya virus, okay, is actually not an anti-inflammatory agent. So they have very different properties just by fine-tuning the structure. And actually, that was our chikungunya work. And we also looked at some targets. It was likely to be a host-related factor. And for reasons, because we haven't really published it yet, we also looked at SARS-CoV-2. And we found that if you took enterographolide and you looked at the inhibitory action against the protease, it was not an inhibitor of the protease. Okay, so somewhere along the line, you know, some of the results need to be checked. Actually, I would say a lot of the COVID-19 literature needs to be checked, because a lot of it basically was published very, very quickly because there was like no other options, right? Okay. And if you think you're going to use enterographolide to treat COVID-19, don't do it, because it's got very low, very, very low antiviral activity, and it's quite toxic. Okay, so you will end up needing maybe a kidney transplant after this if you keep ingesting a lot of enterographolide. But through our structure activity relationships, we actually found a compound which is more potent, which has a completely different target. So this is something that my graduate student is actually, he's writing up his thesis. So hopefully we will see this paper pretty soon. So the question is really, what does the future hold with natural products, right? Firstly, you need to know that not all natural products make good drugs, but it is actually possible to use natural products as the starting point in drug discovery. And if you think about the polypharmacology, right? If you have compounds that have been reported for different sort of indications, then it actually should be possible to design and fine-tune the structure, the chemical structure of the compound, so you would be able to use it to treat specific sort of diseases. And that, I think, is kind of really cool. So it's a little bit like this, right? You can see in some films, okay, there is like a remake. The remake is like not exactly the same as the original movie. So it's a little bit like this. For a lot of natural products, you can start with the original, but you modify the original structurally, okay, to be able to give you what I call a nature-inspired drug discovery and development. So in my very last slide, I'm just going to say that I, for one, am convinced that there is so much that we don't know, okay? Because if you read the literature, you would see everything seems to be discovered. But the reality is that you need to be able to sift what is real and what is not, and by asking questions and by investigating them again. So that's all I wanted to say, and thank you very much.